Could you do me a favor? Could you do me a favor? Would you log on to the Zoom channel, like on Canvas, is the Zoom link, and see if it's streaming properly? That would be super helpful. Does it look okay? Let's see. Okay, cool. Thanks. Will you leave it on while I adjust this? All right, what's it look like now? Okay. It looks like I'm up now. Just me in front of the slides. Okay, thank you. I think that did it. Yeah, sure.
I'm not gonna be able to attend your office hours because I have work, so I just wanted to ask now. I was wondering, what exactly about the taxonomy did you want us to memorize? I wasn't quite clear on that.
Just that first table that's in the syllabus.
Okay, so
Know the characters of each of those fishes, and then the common names of the groups of fishes that are
okay, gotcha. So like
ordered.
Okay, so know a couple examples of fishes that are in that.
To be able to describe it.
Okay.
And I basically went over those on the diversity.
Okay, I just wasn't like quite clear on like what the memorization aspect.
That's the level that you should know it.
Okay.
I'll come back.
Okay, thank you so much. Yeah, I wanted to go to office hours, but I realized that I had work during the entire time. So
Yeah, no worries.
yeah. Oh, thank you so much.
All right. Okay. Presumably I'm audible. Okay. So where we're going. So for Friday, you should have done quite a lot of reading from the textbook on anatomy and physiology. I know that there were many chapters that I assigned for this week. And again, I encourage you to read the figures, read the boxes. If you don't understand something, either you'll pick it up in lecture or you go back into the text and figure out what you need. So you're not reading hundreds of pages. The stuff that's relevant is the stuff I'll be talking about in lecture. So if I mention in the lecture, it's fair game. And if it's not mentioned in the lecture, it's not going to be, you're not going to be required to know it. So a little more reading, pages 94 to 105, plus chapter 8. I believe chapter 8 is on feeding and reproduction. And that'll be important for Friday and possibly for Monday, depending on how far behind I get in today's lecture. I'd like you to read the draft of fish paper for Friday, because that assignment comes up essentially. You're responsible to start thinking about it as of Friday. And I'll spend some time explaining what I mean by this paper and the process of writing a scientific paper, and hopefully giving you some solid tools. And then next Wednesday, Ruth Gustafson from the library is going to come and talk about extra tools and resources that are available there. And I think that's going to be a really valuable thing. even if you've made progress on your paper, just in terms of dealing with the literature, dealing with the search engines, dealing with the citations. I think she's just like this well of information. And for the rest of your time here at UC Davis, if you meet her in this class, you'll be able to walk right up to her desk and strike up a conversation, and she will help you out. She lives for that, as it turns out. She loves to help people. So I think that'll be a great resource. And then you'll submit those papers. This is going to be a draft that's going to be due, I think, October 13th. It's in the syllabus. And then you'll do peer review. You'll basically review other people's papers. You'll submit it back. And then the final will be due later in the month, the final paper. So what you can do to prepare for that, in addition to reading that draft, that draft of fish paper in modules for this week, pick a fish. You'll also see a link on that assignment page that'll take you to a Google spreadsheet, and there'll be a list of like 150, 170 different fishes, which are sort of wedged into the taxonomic chart that I asked you to know for this class. So I've expanded that beyond orders to families, to genus, species, to individual common names. So pick a fish. So if you want to do that, if you have a specific fish that you really hung up on, cool. Get in there and sign up early. If you have a fish that you really want to do and it's not on there, go for it. But here's the thing: is no double dipping. Everybody chooses one fish. And so if somebody's taking it, you got to choose something else. Don't bump them, please, or else we'll just lose the fish. Okay. We'll have a participation assignment today. Give you a little bit of time in class, but please finish it before midnight. It's really easy. It's non-graded. It's just for you to think about and prepare for the test, and you get 5 points for it. Okay. Please stay up to date with it. I think participation quiz is like 20% of the grade. And speaking of quizzes, there will be one next Wednesday, October 6th. It'll be like short answer, fill in the blank, multiple choice, that sort of thing. Any questions about our trajectory forward?
Anything that we've learned thus far is fair game for the quiz?
Yeah, everything through next Monday will be fair game for the quiz. And obviously I'll put an emphasis on stuff that we've learned up through this Friday. But yeah, anything for Wednesday. Yes. Yeah, the quiz. Oh yeah, thank you. The quiz will be on Canvas. So you'll need your laptop, or if you can do it on a phone, you can do it on a phone, but the laptop's probably better. I'll give you like 10 minutes to do the quiz in class, just to make it, just to give you time. But you'll have till midnight to work on it, to submit it, I should say. So you'll give you 10 minutes. You can talk to people about the questions. Well, the order of questions are going to be different for everyone, and you're not going to be able to go back. But you can't discuss the questions here in class or after class. Everything is open book. The idea is process, not product. And yeah, so you'll bring a computer, be ready to take advantage of it's going to be like 10 questions at the most. It's a minute a question. You should be able to finish it. If not, you'll finish it that night. Okay. Any other questions about that process? Yes. I try to do that. I know many of you have requested that, and I'm trying to honor that. As it turns out, I'm cranking on the lectures like right up to the last minute sometimes. And so I could post rough drafts, but it turns into a lot of posting for me. And I'm writing the class as I go along because I never taught it before. What I could do, if you really want, I could give you access to Andrew Ripple's lectures from last year's. Actually, I guess Andrew would have to do that. Maybe I can't do that. I will try my best. Hopefully, I'll get a little bit ahead of the curve and be able to post some things, but I'm often changing stuff. And that's when I'm not. So if that's really buggering you up, we can talk and figure out something. But I'm doing the best I can, I promise. Like I was writing this lecture until late last night and then into this morning just to add things. Yes. Sorry, I couldn't hear. Just the slides? Yeah, I do. Yeah, they should be up for all the lectures so far. And in fact, this lecture is up now. So I usually do the lecture posted as a PDF, and then there will be also a video copy of it as well. At least there should be a video copy. Of course, I'm struggling with the technology, but I think as of today, maybe it'll work. We'll see. You can tell me. Any other questions about that? Okay. And if you have an issue with, I welcome feedback. If something's not working for you, let me know. I'm not terribly fearsome, and I actually want the class to be fun and to be challenging, but I don't want it to be torture. You know, whatever I can do to facilitate your experience, I'm happy to try, as long as it doesn't kill me. Okay. All right, onward. There's this thing called the TSC, what is it? The Science Coalition. I don't know what the T stands for. It's a student video challenge. I'm gonna play this video here for you. So real quick, it's like a scholarship or there's an award for like the best video. And UC Davis has come into, like last year, there were like 3 or 4 students who submitted a video and they placed, you know, they did really, really well. And so the campus is pushing it because it seems like we have a reservoir of interest and talent in this. And so I was thinking that if a team of you wanted to do this instead of the blog, that would be cool. The only problem is we just found out about it and it's due October 18th. So that means you'd be writing the paper and doing this video. And this is like a non-trivial thing. It's not like you can go up and show like cue cards and like, you have to have like production values and stuff. But the campus has some people who want to provide resources. If you want to do it, talk to me. If you really, really want to do a video instead of a blog, then we could talk about that. That's been done in the past, but it's been problematic because unless you're talented, unless you have a skill, it turns into more work than just writing a few paragraphs for blog. But I'm going to play this for you to inspire you anyway as a PSA to the campus.
Coastal ecosystems are changing rapidly in response to ocean warming, extreme events, and other disturbances. But to understand future changes in marine life, we need to document what biodiversity is currently present. And even here in California, the distribution of many marine invertebrates is still poorly known. Therefore, there is a critical need for assessments of biodiversity, which will provide baseline data to evaluate future changes. One example of a group of marine animals that we know surprisingly little about is nemerteins. These unusual worms often have striking coloration and a long, thin body without segments. My name is Maddie Fry, and I'm an undergraduate researcher at UC Davis. Because these animals have been so understudied, there are a ton of unanswered questions about their biology. The goal of my project is to record as many species as possible in the Bodega Marine Reserve. To perform this work, we spend a lot of time in the field during low tides. We can find some of the larger ribbon worms by looking carefully in the field. We have also collected many types of habitat. I then sort through these samples in the lab to find nemerteins hiding in the different materials. Then we document the worms and sequence their DNA. So far, we have identified nearly 40 species in the Bodega Marine Reserve and surrounding areas, including several species that are new to science. This project has shown me how much exploration of marine diversity remains to be done.
So that could be you. Yeah, obviously high production values. Can you pull that off in 3 weeks? An honest answer. If you can, and you have something to do on fishes and you want to go for it, then that takes the place of your blood. Just offering it there. But come talk to me before you jump into it if you want to use it for credit for this class, okay? All right, yesterday's or Monday's question, participation question. I let you guys think about it and put it in an answer. And I just wanted to know what your responses were based upon the body shape of these fishes, the type and placement of fins, spines and rays, their color, the size of the eye. What can you say about their history or their life history? So real quick, what do they eat? Or how do they eat? Any ideas? Yes.
I was thinking of the top fish was kind of, well, obviously it's living. And then I was thinking it's kind of like a fast ambush predator based off of the length of its fins. And then
What do the fins tell you? Why do you say that?
You pull it up real quick. I already forgot.
Oh, just wing it off the top of your head. What do you think? Somebody help her out. She's on the spot. Thanks, Amelia. Yes.
It's because it has the curve, the arrow, the tail knows that it's not.
Yeah, check it out. Yeah, the sort of deeply forked caudal fin means like power. It also has a second dorsal, like a really large second dorsal fins in the posterior part of the body. So it's when it flicks that tail, it really moves fast. Ambush predator. As opposed to this one, what would you say about the shape of the tail and the placement of the dorsal and anal fins on that? Yeah.
So I was going to mention on how it eats through like bubbles and like the how it ate up the bubbles and melting underneath, like probably bottom feeder.
That's right. Yeah. The mouth on the sturgeon, maybe you couldn't see that, but the mouth's down here. So it's a, what's that? What do we call that mouth? Inferior, yeah, inferior. And the mouth on this one is terminal. And if it was above, it would be a top feeder, superior, right? So yeah, that's a real difference in terms of feeding. Ambush predator, the mouth is forward. That's a acanthopterygian fish, so it has this weird mechanism of opening up its mouth and then extending these bones and like creating suction. It's pretty weird. We'll learn about that. And this is just slopping around for poop on the bottom. So good point about feeding. And then about propulsion, this one has what kind of a tail?
Asymmetrical.
Asymmetrical or heterocircle, good. And so, yeah, it has a heterocircle tail because of its ancestry, but you can see also it's developed some extra propulsion here from the posterior placement of the dorsal and anal fins, which probably means it's going to be in what kind of a habitat? Currents, yeah. So sturgeon, although sturgeon often are found on coastal ecosystems and in the bay and in the delta, they tend to be in the deep channels where there's a current and they move upriver to spawn. In order to do that, they need to have some mechanisms to be able to propel them. And so the dorsal or anal fins have come in to support that weak heterocircle tail. So we know that has some aspects of living a riverine lifestyle. This one's more of a reef fish. You can also tell by body shape, right? The kind of disc-shaped, perciform shape on the top one versus the elongate. I wouldn't call it exactly tuniform, but it's more hydrodynamic, right? Any other thoughts about this? What else did I ask? Where in the water column do they live? Well, I think that's pretty clear. What kind of flows can they handle? Habitat's the other thing you can tell about these fish based upon the eye and the color. What could you tell about the quality of water they live in? Yeah.
So swirl fish kind of fish has just like word of life. It doesn't have like little light, but not like extreme, like but not no light. So like it uses like large light fish to detect light.
Right.
Like this sunlight area, so like all year round light.
So probably clear water at depth.
Yeah.
Right?
Yeah.
As opposed to this fish, which would be super muddy water. Super muddy water. It's got very small eyes. It doesn't rely on them a lot. It has a really sophisticated lateral line and a series of pores that help it detect current, which you can see and you could see if you knew that. It's also got these barbels here that help it detect stuff on the bottom. It doesn't rely a lot on vision. It relies on other senses. Yeah.
In regards to the question about the presence of the rays and the spines,
Yes.
fish that both are present, or I'm assuming the rays for
They are. This is a Kondrskian and this is a sturgeon, so it's just got rays. It's an ancestral fish. That fish up there has both spiny rays and soft rays present. So, and people have asked, just to clarify these. Whoa, wasn't supposed to do that. Why are you doing that? Sorry. Okay, so these are the spiny rays right up here in the front, and these are the soft rays back here on the second dorsal. There might be one spiny ray in the front, and then there's a spiny ray here followed by soft rays. Same thing here. The difference is, people have asked about this. The spiny rays are round and sharp and hard. They're unsegmented and they're very stiff. And the soft rays are segmented. They have little chunks. They articulate, and they're very flexible. So they provide some resistance or stiffness for the fin, but they can also flow with the water current. These spiny rays create a much more drag. They have muscle control over them, so they can lay them down if they want to shoot forward. They can prop them up if they want to stop or if they want to turn. So it gives them a lot of fine control. Does that answer your question?
Oh, well, my question was just gonna be, sorry.
No, it's all right.
It was just gonna be like, because it's present on that first top fish, does that mean that it's living kind of like an environment full of predators, and the lack of them on the bottom one would mean there's a lack of predation, or?
Oh, in terms of defense, yeah, I would say that. There's not, the only thing that I know of that'll feed on sturgeon, well, baby sturgeon will be fed on by like straight bass or big predatory fishes. But sea lions will chow on them. But so they're an ancestral fish. So their predator resistance mechanism have to do with the bony plates and these scutes, the bony scutes along the sides, along the dorsal, and also along the ventral side. So they're armored up with a different kind of adaptation. But this fish's spines, essentially, that fish can flare them out and create a much larger area. So the predator has to have a gate to be able to wrap around the spines to get it in its mouth and down its belly. And so that's the adaptive mechanism. So yeah, I think y'all have the idea. Yeah.
And besides the eye, can they, because I said that the top fish might get at nighttime.
Could be, yeah, that's right. It could be it's nocturnal. So maybe it's shallower reef, but a nocturnal fish. That's a really good point. Yeah, or it could just be a depth, like deep clear water depth, but it's clearly visual. And the eye is designed to take into account. It's also colored, right? So they probably use coloring as a kind of signaling amongst interspecific communication. Yeah.
This could be wrong, but I also said like the eyesight is relative to like them spotting predators and trying to like escape them.
Absolutely true. Absolutely true. Their eyes, really large eyes like that might be, depending on the array of rods and cones inside, might be just designed to detect movement, right? Other points? Yeah. Yeah, so they have different styles of movement. So essentially this fish is a slower moving fish, but it's used to dealing with at least periodic high velocity currents. So it probably has a lot of what we would call slow twitch muscle, like red muscle mixed with white muscle. We'll talk about that a little bit today or tomorrow. And they're used to kind of plugging along. So when they hit a ripple or a seam, they've got enough power to be able to bolt through it. But they're not going for speed, they're going for endurance. Whereas this fish up here, that fish can dart. That can just kind of disappear in a flash if it needs to. So it's all about fast twitch or weight muscle. Yes? I would call that, what would we call that? It's like it's a similar body shape to the northern pike that we saw. What would you call it? Fusiform? Yeah, it could be fusiform. Yeah, the ambush predator. I wouldn't call that. I would call that compressed. Yeah, typically, right? Yeah, I think in this case there's a bit of overlap here. It's not really an ambush predator, but because it feels like, so that fusiform body is ideal for ambush predators, but it's a really good point, not only for these benthic animals as well. And the sturgeon has also got a very flat bottom, so it's a little bit depressed. It's a little bit fusiform, a little bit bummed out, a little bit depressed, right? So, stupid joke, but yeah, it's one of the fishes that it doesn't fit neatly into those categories. But the thing is, like with a fusiform fish, it's somewhat hydrodynamic. I think that's the point. It's used to dealing with currents. A more hydrodynamic version is the tuniform morph, and those fishes are adapted for sort of endurance swimming at high speeds through variable environments. So they're even more streamlined. I hope that answers your question. These things are not exact. These are descriptions. And of course, nature is much more complex than a few words. The array of adaptations in these animals are pretty broad. So we try to categorize things. It's an imperfect process. Okay, I'm going to move along. So I wanted to come back to something that I felt like I glossed over because we're running out of time. And that's the length-weight relationship. So length to weight, it's a ratio of the weight divided by the length, and it gives you this great number, this index of how fat the fish is compared to its length. And so what we, in fisheries, we use this to assess the stability or the health of stocks. And people over many years, through across many studies, have developed these length to weight ratios, these relationships. As you can see, somebody fit a curve to a population. This was a population of bluegills. And the equation at the top describes the shape of that line. And the R squared value is a measurement of fit. And 0.99 is like a really bomber fit. Like you don't get 0.99s in ecology, but that relationship is pretty intact. So for most populations under standard conditions, you could expect, a fit like that, say for this population of bluegills. But say something's changing. Say agricultural practices are changing around the pond. Say climate change is heating up the lake, stressing out the population. You could go back after this relationship's been established and do the same kind of study. And if you found that the length, the weight to length ratio, the condition factor has dropped, and you can start to say something about how stressed out that population is. And then you could conjecture about what factors are affecting it. Well, clearly, probably there's some food limitation, or maybe if it's getting hot, they just can't eat enough food to continue to burn the calories they're burning because of the heat, because they are mostly kilothermic. So the relationship is very useful. And just in short, these are three different individuals from a similar population. And the fat one up at the top, tends, a fish like that would be up above the line. It would be a little bit higher than average. And a fish, that's really skinny like this would be below the line. But together, the trend makes up a whole population. There's always going to be intra-population variability of individuals, but then there's going to be population change as a whole. And of course, the goal of a scientist is to say, well, if the population's changed, how much has it changed? Because individuals behave randomly within a population. And that's why we use statistics, so that if we had a new line that developed here, or we thought we had a new line that developed, you could use statistical analysis to try to say there's a 95% probability that these populations are similar, or a better way to say it is there's only a 5% probability that these populations are not different. Okay, we're not going to get into stats and class, but this is an example of the sorts of things that fisheries managers use. And I wanted you to be familiar with this idea of condition factor. So back to external anatomy, there's a few points that I wanted to address that I felt like I'd missed. Actually, somebody asked me about the spiny dorsal and the soft ray dorsals, and so I pointed them out here. I think I addressed that earlier, so I'm going to move on unless anyone else has questions about dorsal or about spines versus rays. Okay, and here I pointed out the series of soft rays. So typically, I do want to make this point, is that usually the spiny rays are in the anterior side of the pelvic, the anal, and the dorsal fins. And part of that is probably because of hydrodynamics, part of it's probably because of steering, and part is probably because they're trying to protect their face from predation, right? It's a defense mechanism. Okay. Some other things that I missed, but I want to bring to light to note is that the bronchiostical rays are a series of soft rays that exist that are basically, they hold up the operculum. The operculum is the gill cover. And the bronchiostical rays form the bronchiostical cavity or the gill cavity. And so they have to be flexible because the gills need to fill up with water to be able to extract oxygen from them. And that's useful to know later. I'll be referencing them. And finally, for external anatomy, something I failed to point out, or maybe just pointed out really in brief, is the lateral line. And the lateral line is a really important sensory structure that if you were in lab, you were starting to look for them. We'll see them in the photos over the next few slides here. And I'll try to explain a little bit more about how this lateral line works. So here on the tule perch, you can see you can see that change where the arrow is pointing. I guess I should go up here and use my pointer. So right along here, this is the lateral line, right along there. And that lateral line is basically, it's like we call it an underwater sixth sense. It's maybe analogous to like the hair on your arm. So you can detect movement of air through that mechanism. We call it, I think we just call it touch. In fish, it's much more sophisticated, but it's built on the same principle. It's mostly an underwater sense. And there are these pores at the surface that open up into a channel that's subdermal. It's lined with these specialized scales called neuromasts. And so here you can see the structure of the lateral line. Now, the typical lateral line that's most visible is along the lateral side of the fish there, just above the pectoral fin and then down towards the caudal fin. But they tend to have these channels, or at least pores with neuromasts in them, all around the head and the eye and the mouth. Why? Because that's the most important part of the body. The brains are up there, the feeding mechanisms up there, smell is up there. They need to protect that part of the body. So it has to be particularly sensitive to changes in current and water movement and temperature. And the way they do it is that these neuromasts have they have this thing called a cupula, which is right here. And inside the cupula, there are all these little hairs, these sensory hairs, and they're kind of embedded in this weird gelatinous material. And as water flows through these pores into the channel, it bumps up against the cupula and it makes these sensory hairs kind of like the cilia in the cochlea of your inner ear. It makes them move. And the fish can interpret that as very subtle water movements. And this is extremely useful for detection of shifts in currents, evading predators, and in poor visibility conditions, fish rely heavily on this. They also use it for schooling. So it turns out that fish that are blinded are still able to school using the lateral line, but they tend to increase the distance between them. And then fish who have sight but lack a lateral line, because scientists have ablated it. It's a little grisly, but it's an experiment somebody did in the 50s. If they ablate the lateral line, that'll decrease the distance. So they can see, if they can see each other, essentially, they'll move in close. The lateral line allows them to detect currents, which keeps them farther away. And it's the combination of those two senses that interact to keep a fish in a school tightly packed in an orderly way and keeps them from bumping into each other. And I'm sure you've seen, if you haven't dove, you've seen on YouTube or, the Great Blue Earth or whatever, you've seen these massive schools of fish and they're all coordinated. Well, they're doing it with eyesight and their lateral line and maybe even chemosensory mechanisms as well. Anyway, that's the lateral line, the neuromasts, a really cool sensory thing that we don't have. Also, the seventh sense that a number of animals have in the tetrapods as well as the fish is magnetic reception. And sometimes that's related to the lateral line and other times it's another mechanism. We actually don't know that much about it, but many fishes do have this pretty sensitive ability to detect weak electrical fields that are generated by the Earth's magnetic field to orient themselves. And this is what allows certain fishes that are migratory, like salmon, tuna, eels, also sharks and rays, which are very, very electrosensitive. It allows them to navigate their migrations. And in the case of salmon, to return back to their natal rivers. So when they go off to the ocean to feed for 3 years, they're able to navigate to the mouth of the river. And then they use sense to get and actually to hone in on their sight. Anyway, these are specialized senses: olfactory, lateral line, and otoliths. And oh yeah. I'm sorry. Yeah, that note there. Yeah. So in terms of magnetic reception, there's some sense of smell maybe tied up in this, and otoliths, which I haven't talked about, which are these inner ear stones, sometimes have a bit of magnetite in them, and that magnetite responds to the Earth's electrical field. So that's most of the outside of the fish and most of the sensory organs that I'm going to talk about. So I want to talk about internal stuff. And the key elements of their anatomy are, check my time here. Thanks. The key things that I really want you to know about, and I'm going to discuss in the lecture, are the air bladder, of course, the gills and the gill structure and functions, a little bit about the heart and the circulatory system, and then later this week, early next week, we'll talk about the reproductive system. We'll also talk a little bit about the digestive system, the stomach and the intestine. There's no great surprises there. It's a pretty simple system. But for starters, let's log into the, or drill down into the circulatory system and discuss that. There's some unusual features of it. They have a very simple heart. It's a very simple circulatory system. So they have it's a 4-chambered heart, but it's basically reduced to 2 main chambers. The atrium and the ventricle are the primary pumps. And blood is circulated through the heart, across the gills, across the body, and then back to the heart. And this is really different than most tetrapods, where with our very well-developed 4-chambered heart, we have a pulmonary vein that goes to the lungs. So blood flows from the body to the heart in tetrapods, most tetrapods, goes to the lungs, goes back to the heart, and then gets propelled forcefully through the pump back to the rest of the body. We have that mechanism because of gravity. Fishes don't need it because they live in a buoyant medium, and the viscous environment that they're surrounded by essentially compresses their body and assists in moving blood throughout the system. And so they don't need a whole lot of force to move blood through the system. And in fact, inactive fishes can have super tiny hearts, where mostly the blood is getting sloshed around when they move once in a while, especially deep water, cold water fishes that have very low oxygen requirements. And the gills, of course, are the primary breathing organ in most fishes. And this is where blood circulates through to oxygenate itself. And the gills, so I already talked about the branchiostegal rays, which form the gill chamber. And then the gills themselves are hung up. They're placed on these cartilaginous structures called gill rakers. And those exist inside and underneath the operculum, which in this picture here, this guy is basically he's got his thumb up against the operculum, and he has his fingers through the mouth of the fish, and he's holding up the gill rakers. And you can see that you can see his fingers. I hope that shows up. Is it showing up? Yeah, my little showing up. You can see his fingers between the gill rakers, right? Makes sense because water is going to go in through the mouth, across the gills, and back out under the operculum. And the branchiostegal rays form a sort of pump, this sort of a chamber. The mouth pushes water through, the branchiostegal rays and the operculum squeeze that water back out through the back. So essentially, if they're not, the thing about the teleostean fishes is that they don't have to be swimming all the time because they have this pump mechanism. The mouth gapes water, pushes it to the rear, and the operculum and the branchiostegal rays push the water out. So they can just sit there and pump. And if you've ever had a goldfish, that's pretty much what goldfish do all the time. You see that mouth working, it's pumping water across its gills, something that the elasmobranchs can't do. So this is the front of the fish over here. This is the front or the anterior of the fish. And water moves from the mouth across these and out along the side of the fish. And the front part of the gill arches, these like white cartilaginous spikes there, those are called gill rakers. They have a different function. They're for feeding. On the other side, the red stuff, it's red because there's lots of blood moving through it. Those are the gills. That's what we're talking about right now. So here's another view of it. These are the gill rakers. This is the gill arch, which are these series of cartilaginous loops that sit within the gill chamber. And the gill rakers are on the anterior side, and the gills are on the posterior side. So water flows across the gill rakers and back off the gill arch. And that's an important thing to remember when we're thinking about gill function, and I'll get to it. So the gill filaments, you can see here, they call them gill filaments or the lamellae. They're these blades, these very kind of thin blades that are highly, they're highly vascularized. They've got lots of blood flowing through them. And that's what you see when you see a gill. And I already talked about how they force water past the gills. And in addition to consuming oxygen, they also take carbon dioxide away. And the gills are also useful in removing other waste products. They're part of the osmoregulatory system for fishes. And then those branches are lamellae. They have this very thin epithelium, which promotes gas exchange. And I'm going to talk a little bit more about that gas exchange. So countercurrent exchange. I don't know, has anyone ever encountered this principle before? Yeah, biology, you've hit it. So maybe my task isn't too steep here. So countercurrent exchange is this amazing mechanism that prevents, essentially enhances the ability of the gills to extract oxygen from the water. And the way it works is that flow through the gills, blood flow through the gills, will go in the opposite direction as blood flow through the water. And what that means is that low oxygen water, scratch that, low oxygen blood that's already gone through the body is going to come in contact with low oxygen water. Well, there's a net higher concentration, even in the low oxygen water. There's more than there is in the blood. And so you're going to have a net flow, although small, you're going to have a net flow into the blood. As the blood progresses towards the water, it's going to come in, it's the partial oxygen pressure of the blood is going to increase, but the partial oxygen pressure of the water is also going to increase. So oxygen is going to continue to flow at about the same rate into the blood. And at the point of contact where you have pretty highly oxygenated blood, well, you're also going to have the most highly oxygenated water. And so you're going to continue to have water. Scratch that. You're going to continue, I'm trying not to confuse you. You're going to continue to have oxygen flowing into the blood. So that means at all points of exchange, you're going to have that transport of oxygen from water into the blood, because the partial pressure of oxygen in the water is always going to be a little bit higher than the partial pressure in the blood. If you had water flowing through the gill in the opposite direction, if you had it flowing this way, then you would have poorly oxygenated blood coming in contact with high oxygenated water, but the process would diminish as they both drift, as the both flows went in the same direction. You wouldn't have a continuous exchange, and so it would allow the blood to capture less water. Ah, how many times can I mess it up? To capture less oxygen. And just in case I really messed you up, I got a video. And maybe this guy can do a half good job of it. So I'm gonna let it play. I'm gonna try not to blow your ears out, okay? Weird. Sorry. I actually hate showing videos because it's so challenging. All right.
In this short video, we're going to look at an example of formulating the function in biology using countercurrent gas exchange in fish gills. So we know that.
Oh my God. No, I'm sorry. I'm a spaz. I knew that countercurrent exchange would mess me up. All right, one last time.
In this short video, we're going to look at an example of formulating the function in biology using countercurrent gas exchange in fish gills. So we know that fish use gills to extract oxygen from the water to release their carbon dioxide. When we look here at this picture, we can see someone lifting up the operculum of a bony fish. We see this gill arch, these bright red gills on there. And these gills are so red because there's lots of blood vessels running through them. And these highly folded, very thin membranes make for a very good gas exchange surface. So what we're going to need to do is have a little X-ray vision to look inside of our fish here to see the 4 bony gill arches that we see right here. And those gill arches are supporting this very highly folded thin membrane of the gills, which I'm representing by these red squiggly lines here. And so we have an opportunity to design or look at the design of how the fish gills work. Water enters into the fish's mouth, moves across the gills, and exits the back of the operculum. And as the water goes through, oxygen is extracted from the water. So I'm going to put some numbers to this just to make it a little easier to understand. I think it really helps. And I've picked the numbers kind of arbitrarily, but it'll make the point. So imagine that the water coming into the fish's mouth has 100 parts per million of oxygen. By the time it reaches the end of the gills, it's down to, let's say, 80. So we've extracted 20 parts per million of oxygen out of the water diffused into the blood. And now I'm going to let you be the bioengineer. You have the option, A, to send the blood into the gills from the front. So we could come in and go through here from the front and exit the back. Or you can have the option of coming from the back, option B, send the blood through the gills this direction and out the front. So the question is, which would you choose? Now stop the video and think about which would be the better option. The problem with this is, you know the name of this video. If you were in my class and I were teaching this, I wouldn't talk about countercurrent till the very end. So knowing the answer is one thing, but understanding it's a whole nother thing. So let's look at the why. So let's isolate the system from our fish, move the fish out of the way, and zoom in and get a little closer. Now, one of the things we have to know is that gases tend to move down their concentration gradient. So if we bring our blood in this direction, choice A, and the blood's going throughout the whole body, so by the time it reaches the gills, it's fairly oxygen poor. So I'm going to pick the number 60 parts per million. So as the blood and the water meet each other, there's a favorable gradient which allows the water or the oxygen to move and diffuse out of the water into the blood. But by the time we get to the end and we've picked up 20 from the water, the blood is at 80 and the water's at 80. So our difference between the two, we could reach an equilibrium. Now, I did pick these numbers at random, and I could have just as easily, or arbitrary, not at random, I could have just as easily made this number 50, and then by the time we gain 20, we'd be at 70, and we'd still have a favorable exchange rate from the 80 to the 70, but we certainly would have a much less steep gradient, and we would reduce our rate of exchange. So let's turn the system around and bring the blood in from the back of the gills and have it run countercurrent to the water. So again, the blood's coming in, we chose 60 for the blood coming into the gills. We have a favorable exchange gradient between the 80 and the 60. Our most oxygen poor blood is meeting our most oxygen poor water, but the water still has more oxygen than blood. And by the time we get to the end, our most oxygen rich blood is meeting the freshest water to ensure a favorable gradient throughout this entire system. This countercurrent exchange gradient or exchange flow maintains the most favorable gradients for gas exchange. So we can put the system back into our fish and we see that the countercurrent, the blood flowing from the back of the gills towards the front and the water flowing from the front towards the back is going to give us the most favorable exchange gradient. And this is a very good example of form relating to function in a biological system. There are many others. For example, when we look at the heat exchanges in the arteries and the veins, and also water conservation loop of Henle. So these are things that'll come up pretty frequently in biology. All right, so if you have any questions, watch the video again.
Okay, so I don't think you want to watch the video again, but you can. Any questions about that? process is really important. We'll come back to it again and again, which is why I want to take a little bit of time on it, because it makes life possible in the water for a variety of reasons. So no participation grade today because we didn't get to my participation credit. We'll start up next week with a little bit more about oxygen solubility and then on to digestion and whatnot. Thank you.